We'll now continue our discussion of cell energy, discussing the details of photosynthesis. So, what you need to know to begin with is, of course, photosynthesis. The word photosynthesis means light production. Photo means light, and synthesis means production or making of something. And so we need to talk a little bit about light and the characteristics of light and how light is absorbed. So you know, of course, that sunlight contains electromagnetic radiation. Visible light is only a small part of that, okay, and we're looking specifically at visible light. Light behaves, even though it travels in waves, it behaves as if it's made of little packets of energy called photons, and that's a fixed quantity of light energy that you find in the different wavelengths or the different colors of light, and we'll talk about the length of the wavelength compared to the amount of energy, of course. Basically, the shorter the wavelength of light, the greater the energy that you have in that light. Here we have a representation of the electromagnetic spectrum, and we're looking at the enlarged portion here, visible light. Visible light goes from violet, 380 nanometers or so in length, all the way up to the red, which is 750 nanometers. You see that these, since we said that the shorter wavelengths have more energy, that means the violet and the blues have more energy than the reds and the oranges. And so we're going to be looking at a couple of different colors of light as we talk about how they're absorbed, how the wavelengths are absorbed by the chloroplast. Pigments absorb light. Okay, and the pigments, chloroplast, and chlorophyll, and other pigments are built into the thylakoid membranes. They absorb some of the wavelengths of light and reflect or transmit others, and that has to do with their appearance. Okay, we see the wavelengths that are transmitted and reflected, which is why plants look green, because they transmit or reflect green wavelengths rather than absorbing them. So you can see here that they absorb the blues and the reds and the violets, but they transmit the green and reflect the green. So that's why leaves appear in different colors of green. Now, we have several different pigments that we find in chloroplast, and they absorb pigments the light in different wavelengths, which is an advantage to be able to use different wavelengths, because sometimes some of them get filtered out. Chlorophyll A absorbs colors in the light in the blue, violet, and red range and reflects green, and the chlorophyll B absorbs blue and orange light and reflects yellow-green. So they're slightly different from each other. There are also other pigments found in plants, and you see this sometimes in leaves that are different colors. One important group of accessory pigments are called carotenoids, and they also assist with light collection, and also part of their purpose is to help protect against excess radiation. They absorb some of those really high energy wavelengths, or absorb some of them to keep there being too much energy absorbed by the chlorophylls, which could be damaging to the tissues. In the chloroplast, the pigments absorb photons. That's how they capture the solar power. And what this does, when they absorb the photons, the potential energy of the electrons is increased, and that sends these electrons into an unstable state. And then as they move back down or drop back down to their normal or the ground state, they're going to release energy as heat. And so what photosynthesis does is it traps that energy in molecules like NADPH and ATP, and then we can use that energy to power the processes that make the sugars. So the chlorophyll pigments and other pigments absorb the photons and transfer their energy, and then we are going to transmit them from one molecule to the next. In the thylakoid membrane, we have the chlorophyll molecules and some other protein molecules arranged into these groups of molecules called photosystems. The photosystems have the light harvesting complexes made of the pigment molecules, and they surround a reaction center complex that acts as they act as a light gathering antenna, so they absorb or attract the energy of light and then use it in this reaction center to produce the reaction. So here we have a photosystem. Notice that we see it in a phospholipid bilayer. That's the thylakoid membrane. Remember, the thylakoid membrane is a regular phospholipid phospholipid bilayer with embedded proteins and other molecules in it. So here's our photosystem. We have the light harvesting complexes made of these pigment molecules, and then we've got a pair of chlorophyll A molecules here in the center that send electrons up to this reaction center complex, which is the primary electron acceptor. And then the electrons are going to be passed from one molecule to the next, sort of like we saw in cellular respiration, in order to power some other processes to make energy. This light energy is passed through a series of molecules. It's an electron transport chain of a type, okay, within the photosystem. And then when it reaches the reaction center, the primary electron acceptor accepts the electrons, and then that's the first step to changing that light energy to chemical energy in those light reactions. There are two types of photosystems in leaves. There's Photosystem I and Photosystem II. And they work together in these light reactions to do different parts of the process. The photosystems are slightly different from each other. Photosystem II, which is actually the first one to be activated, is called P680, P for photosystem, because the pigment in Photosystem II absorbs light with a wavelength of 680 nanometers. Photosystem I, which is the second one in the process, is called P700 because its pigment absorbs light with a wavelength of 700 nanometers. So slightly different wavelengths activate these two different photosystems in the leaves. In the light-dependent reactions, the light is going to be transformed into chemical energy in the form of ATP and NADPH. So what we're going to do first is we're going to remove electrons from water. Those electrons are going to be passed from the photosystem II to photosystem I. They're going to be accepted by NADP plus to make NADPH, and then the electrons are going to move down an electron transport chain to provide energy to make ATP. So we have two different molecules that we're going to be making here in the light-dependent reactions. So here's what happens. The light strikes the light-collecting molecules in photosystem II, which are going to activate P680. This is going to give energy to split water molecule. We're going to release the oxygen, and the the electrons are going to be used in accepted by the primary electron acceptor, and the protons are going to collect here in the thylakoid space. Out here is the stroma. These electrons are going to be passed through a system of proteins in the membrane. These electron transport molecules are going to provide the energy to pump protons from the stroma into the thylakoid space. So we're going to have an accumulation of hydrogen ions in this thylakoid space. These electrons are then be passed to the P700 in the photosystem I, which has also been activated by light, and it's going to replace the electrons that have been removed from this, and these electrons are going to be passed over to another molecule, and this molecule is NADP reductase, which makes NADP. It takes those electrons and some protons that are here in the stroma and produces NADPH, which is going to provide energy along with ATP for the Calvin cycle. The products of the light reactions are three things: NADPH, ATP, and oxygen. The oxygen is a byproduct. It is a waste product of this process. It is released to the atmosphere, and the NADPH and ATP will provide energy to make the sugars. Now, we talked about in cellular respiration the process of chemiosmosis, and we have a similar process occurring in the chloroplast as well. It's still going to be chemiosmosis. It's the same mechanism that is involved with the oxidative phosphorylation in mitochondria. In the plant, in the chloroplast, we're going to call it photophosphorylation because we're using the energy from light rather than the oxidative collection of the electrons. So we have this electron transport chain, and what that does is it's going to produce a concentration gradient of hydrogen ions across the membrane. Back here we said the hydrogen ions would be collecting in the thylakoid space. These proteins in the thylakoid membrane will be get energy from the electrons to pump hydrogen ions from the stroma into the thylakoid space where they'll collect. So in photophosphorylation, we're going to use the energy from light to pump hydrogen ions into that space, and then we're going to have a concentration gradient building up, just like we saw in the intermembrane space in the mitochondrion. And there's also ATP synthase there. The concentration gradient is going to drive the hydrogen ions back through ATP synthase, and that's going to produce ATP, just like we saw in the mitochondrion. So here's our two photosystems, our electron transport chain pumping hydrogen ions into the thylakoid space. The hydrogen ions collect here. They build up a concentration gradient, high concentration here, lower here. They're going to pass back through ATP synthase back out to the stroma. So we're going to make more ATP here, and then the NADPH produced here and the ATP produced here will go to the Calvin cycle to give the power or the energy to make the sugars. So as we said, the ATP and NADPH will give the energy or the power to make sugars in the Calvin cycle. This occurs in the stroma, which is the location of the enzyme systems that are needed for this series of reactions. The ingredients that we're going to need are the carbon dioxide from the atmosphere and the ATP and NADPH from the light-dependent reactions. These are going to produce a 3-carbon sugar called G3P, and then the plant is going to use G3P to make glucose and other kinds of organic molecules. So our inputs will be carbon dioxide, ATP, and NADPH. Again, we have a series of reactions here. We have an initial compound that's going to accept the carbon dioxide, and we're going to add one at a time, rearrange molecules, and our output will be G3P, and we'll regenerate that starting molecule, similar to what we saw in the Krebs cycle. So the steps will include carbon fixation, which is putting the carbon dioxide molecules together, reduction, which is going to be adding hydrogen ions to molecules. We're going to release G3P, which is the starting product, the starting compound to make the sugars, and we're going to regenerate the starting molecule, which is called RubyP.